Hello.
We're going to start with you because you had a question that I put off last time about the presentations. So just be ready for that when everybody gets on.
Okay.
Jackson, are you coming to us from a car?
I am. I'm about to walk up to the PLC.
Okay. Okay, Mary, you had a question about the genetically engineered organisms presentation from last time?
I just wanted to make sure. You just wanted like a summary, right?
Yeah, so the whole reason for us doing these presentations is to, for one, well, they have a couple of reasons. One reason is to get a good summary out to all the people in the class. So I want all of you to read all of the chapters, but it's a lot to read, as you've probably discovered. And so I want to make sure that we're kind of all on the same page. So as you present your group's presentation, it will become apparent to all of us, I think, what's understood well, what's not understood very well, and we can use those presentations to clarify issues that are in the chapters themselves. The other thing that it does for all of you is it gives you practice of taking the information in there, which is technical. It's written for other scientists. It's not really written for the broader public, and getting you ready for your paper that you have to write as we approach Thanksgiving, where the idea is that you're going to be writing this to a family member to explain to them what the various issues are with genetically engineered organisms and how concerned they actually should or shouldn't be. And so, yeah, it is a summary. But so, for example, I forget which group you're in and which chapter you have, Mary.
We had chapter 6.
Chapter 6. So chapter 6 is the socioeconomic chapter, is that?
It's the socioeconomic effect.
Yeah. Okay. So for your particular chapter, I think one of the things that you guys should be doing in that chapter is essentially talking about, well, what are the effects of these genetically engineered modifications to the plants in terms of the bottom line of farmers, for example. And that could be that these genetic modifications might increase yield. If it increases yield, well, what does that do to the economics of the farmer? But one of the things that these things might do is actually reduce the amount of time that farmers have to spend farming. So, for example, if one of the modifications is a modification that makes the plant immune to herbicides, now the farmer can just spray herbicides on his fields with something like an aerial sprayer. Just hire a guy in a crop duster to spray herbicide on his fields, which gets the field covered very quickly, rather than going through and repeatedly tilling that field with a tractor to get rid of weeds. What that does is it frees up the farmer from a significant time investment in that crop, which then might allow that farmer to go and seek off-farm income. And so hopefully this is something that you read about, which is not something that the normal person would think about. Oh, well, these genetically engineered crops allow farmers to seek off-farm income, which may make their overall operation more profitable, not because they're getting increased yields, but they're just spending less time farming and they can go do something else with their time. And so this complication and nuance is important to grasp. And so you need to think about what are the genetic modifications that people have made in genetically engineered crops. What are the effects of those from agronomic perspectives that you see in an earlier chapter, but it's things like yield and things like weed suppression. Well, what are the economic consequences of those things? One of the negative economic consequences of those things is that genetically engineered crops have a premium price placed on them, so they cost more to buy. George Naylor from the book The Omnivore's Dilemma doesn't plant genetically engineered crops, partly because of the price premium that is placed on those. He doesn't want to pay that extra price. So the idea is to summarize it, but you need to order it in such a way that it makes sense and is digestible to your colleagues, because you're also going to have to do that in your paper. So think of the presentation as preparation for the paper that you're going to be writing. Does that help at all?
Yes, it does.
Yeah, when I write papers for publication, I generally do the work and then I'll try and present it at a meeting in the summer, and then I'll try and write it up over the following fall while it's still fresh on my mind. So I've always used presentations as a preparation stage for sitting down and writing something, because the presentation forces me to organize my thoughts in a really concise way, and then I carry over that same structure to my writing typically. What else? All right.
I have a question.
Yeah. Blake.
For the paper, you said you have to write it to like a relative, like a family member. And for our Thanksgiving, obviously we are not obviously, but we won't have any other family members besides my close family at Thanksgiving. Yeah. So would it just be to like, could I just write it directly to them, or would I just write it to my mom and dad?
Imagine a not very well educated uncle joined you.
Okay, so you just imagine.
Yeah. So so I'm sure you have an uncle. Do you have an uncle or aunt? Yeah.
Yeah, I do.
Imagine your most curmudgeonly uncle that you can think of. Write it to that guy.
Okay.
Like if I was writing this, let's see, I would probably write it to my uncle Tommy, my father's brother. So you're writing it to somebody who doesn't have a background in science, who doesn't have a scientific education. And so the idea is to get the information across to them, make sure that it's accurate, but make sure that it's communicated in a way that regular people would understand it. The fact that you're getting a college education and that I'm about to load some serious science on you makes you different from most of the rest of the educated public out there. At least I hope it does.
Okay.
Is that
Thank you.
Clear?
Yeah, thank you.
Yeah, it doesn't have to be written to a person that you're actually sitting down at Thanksgiving dinner, but just imagine that you had your extended family there and this topic came up. You know, somebody was watching Fox News or somebody else was listening to NPR and they heard about genetically engineered foods and they had lots of misconceptions, and your job is to write a document to them that would clarify any misconceptions that people might have about this. Genetically engineered organisms have their positive points and they have their negative points, and that's the way with practically anything in life. And so yeah, you need to get across that complexity and nuance. One of my beefs in the year 2020 is that we've thrown complexity and nuance out the window. And I feel that one of my tasks is to get that back into at least your guys' minds. Other questions? Okay, let's finish our conversation about what, hold on, I just got a message. Oh yeah, so the other thing is this group presentation is a group presentation. So if your group members have been contacting you and you have been basically a ghost, you need to unghost yourself. Each of you is going to have to take a portion of the presentation. So I think there are three groups, 4 in each group, but 3 in one group. So you need to divide up the tasks of doing the presentation evenly amongst you. So when you make your presentation, one of you will be sharing your screen, but all of you will be contributing to the presentation, and you're going to be graded as a group. So one of the things that you need to recognize is that your group grade is going to be kind of dependent not only on the best person in the group, but also will be dependent upon the worst person in the group. And so all of you need to bring your best effort to the task, because you don't want to be dragging down your group. So if your other group members have been trying to contact you and you've been non-responsive, you need to become responsive, because it is a group presentation. It isn't something that everybody's going to prepare and one person is going to present, or that one person will prepare and multiple people will present. It will become very obvious to me and everybody else if somebody prepared the material and then all you're doing is reading off of the screen. Now, this is the second year that I've done these presentations, because we've done the report in the past and I wasn't happy with the performance on the report necessarily. So I started using these presentations as a way to get you kind of equipped. Let's see, I wonder if I have those. I should have thought about this before this conversation, but here we are. I might have last year's presentations. Yeah, I don't think I archived them anywhere. I'll try and find them. So last year, I guess I hadn't been clear enough with people about kind of what my expectations were. And the first presentation, which was on chapter 4, which is the agronomic and environmental impacts, it was a group of people standing up in front of the class. We did this in person, and their slides were basically just like really small font, lots of sentences, and the sentences were all just basically direct quotes from the report itself. And so it was basically people standing in front of us reading things that, quite frankly, we could have read ourselves. And I had to stop them partway through, and then we basically had a long conversation among one another about what an effective presentation is and things like this. And so I sent them all back to redo things. And so we put off the presentation a little while, and they went back and they revamped things. And the second go around was much better than the first go around. So don't just quote don't just have a bunch of really text heavy slides quoting the findings of the report, because that's boring. We could all just go read the report, and we are going to all go read the report, but your goal is to kind of make the points that they have to make. And the fact of the matter is, you can in many ways make those points via figures rather than text. You might want to have some text that summarizes things to just kind of drive the point home in text. But in many cases, you can make these points using the figures that are actually in the chapter itself. So make it visually engaging, make it not too text heavy. I had a student doing a research presentation last week, and he sent me his slides, and he said, how do the slides look? I'm like, well, the slides look fine, but it all depends on what you're going to be saying while those slides are up as to whether or not those slides are effective. And we didn't practice. We didn't have time to practice because he got slides to me late. And as a result, he could have done a much better job if we had had time to look at the slides and actually have him do a practice presentation where I could hear what was coming out of his mouth while the slides were up there. And so one of the things that you also probably want to do is practice before you get to class on October 19th. So not too text heavy. The things that you say are important while a slide is up there. And the visuals that you present should be informative and engaging and I guess not overly complicated if possible. And once again, you should present in language that is approachable to the rest of your classmates. You're not making this presentation to me. I've read the National Academy of Science report every year a couple of times since it came out. I know what's in there. You don't need to make this presentation directed at me. You need to make this presentation directed at your peers. Is that clear? All right, I need to respond to a comment here on chat. So just give me a sec. One of your colleagues reminded me to think of something that I forgot about since we last talked. Okay. All right. Other questions? All right. It seems like most of you are still good with the Zoom meeting thing. Is that correct? Okay. All right. Then we'll continue with this. As it turns out, one of my colleagues that I interact with a lot is suffering symptoms that may or may not be COVID. He is getting tested today. And I got 2 notifications on students this morning who are having to go into quarantine. So the thing that I feared might actually be happening, which is that we might be suddenly getting an uptick in COVID activity on campus. I hope that's not the case. Trust me, I hope that's not the case. But depending on what my colleagues' test results are, I myself may actually have to be going into quarantine, which is going to make teaching labs in biology really, really difficult because those have to kind of be face-to-face. So stay safe, keep your masks on, avoid people. Jackson, if you want to take class in your car, that's actually not necessarily a bad thing. So I'll keep you apprised as to what my own status is. I feel fine. I don't have any worries. I basically don't interact with hardly anybody except the occasional student who comes to my office, which, by the way, only one of you has come to my office so far this semester for any substantive help. The door is always open. I will answer it. It may take me a minute to get my mask on in order to answer it, but I'll generally shout through the door, give me a minute. If you're having questions about the material, you can ask them in class. If you're having questions about the material, you can come to my office hours. There's a sign posted on my door that has all my office hours, and the office hours are also in the syllabus. So by all means, don't get to the next test with any sort of misunderstandings of the information. And like I said, I was saying yesterday to a student, there's a colleague of mine at the University of California in Dominguez Hills who stopped calling them office hours and refers to them as student hours. And I keep telling myself that I'm going to adopt that same language, and then I forget to adopt that language. So those office hours are here for you. I'm sitting in my office, and if you're not here, I'm just getting other work done. So use those times. And if you don't feel comfortable coming into the office, I can also do office hours over Zoom. So last fall, actually, back in August, I got this whiteboard installed in my office so that I could have it for office hours. And then when we went to online teaching, I was so glad that I have a 4x8 whiteboard in my office. So avail yourselves to the person on the campus who knows the most about the material that you're learning in this class, which is me. I can say this for sure. Nobody at Williamsville College knows more about this material than me. So, take yourself, take full advantage of that. That's all I really have to say about that. And like I said, I'll keep you apprised as to what my own quarantine status is. All right, you guys watch the news, listen to the news, listen to NPR, CNN, Fox. I don't know which news outlets those would have been on. I heard about this on NPR. Did you guys hear about the Nobel Prize in Chemistry that was awarded yesterday? No, nobody? You guys should really follow the news. I'm going to share my screen. What's that, Blair?
It's all been sad recently.
It is sad. I'm sorry that it's sad, but it's the year 2020. My hope is that the year 2020 will eventually go away and things will be better after the year 2020. Where did it go? Oh, well, let me do this also. So keep up with things that are on the Moodle site. So here we are in this week. I asked you to read chapter 26, section dealing with nutrient cycling, because that's what we're going to start talking about today. Also, this may not come up on Friday, but it will at the beginning of next week. Chapters 8 and 11 in the Omnivore's Dilemma deal with a place called Polyface Farm. Chapter 8 is an introduction to Polyface Farm, and Chapter 11 introduces you to the idea of food webs and nutrient cycling on Polyface Farm. So we're not taking the book, the Omnivore's Dilemma, linearly. We're going to be jumping around as topics come up. So Chapter 8 and Chapter 11 will be useful to you. You guys can see my screen? Is that correct? For some reason, I can no longer see you guys. Oh, there you are. So you can see the screen?
Yes.
Yes, okay, yeah, I couldn't see you respond. All right, so that's what's going on. The exam, let me pull up the schedule. The exam has always been scheduled for the 16th of October, and then right now the genetically engineered food presentations are scheduled for the following Monday. So just be aware that that is coming. Nobel Prize in Chemistry. Two chemists, two scientists, in an unusual move by the Nobel Committee. They happen to be both female. Males dominate Nobel Prizes in all fields. And this is for the development of CRISPR. So CRISPR was discovered in 2011, 2012. And here, just a mere 8 years later, the two women who developed the method of genome editing are receiving the Nobel Prize for this technology. On NPR, there's a nice little article about the awarding of the Nobel Prize. So something that we just went through in class, which is this genome editing technique called CRISPR, is now back in the news again because it's been something that's transforming the field of genetics and molecular biology. And so they won the Nobel Prize yesterday. All right. Nutrient cycling. So in the second part of the book on the omnivore's dilemma, Michael Pollan talks about this place called Polyface Farm. And Polyface Farm is this farm out in Virginia. And they do a form of rotational grazing there that is unusual in modern American agriculture, although it's kind of taken hold in some segments of the farming culture in the United States. I know a farmer who uses this same strategy of growing meat out in Richmond. Back when we had a lab in this class, we would go and visit this farm, Parker Farms Natural Meats. And in the chapters dealing with Polyface Farm, they talk about kind of how Polyface Farm works ecologically. But even before we get to the Polyface Farm section, in chapter 2 of the Omnivore's Dilemma, talking about the farm, we get to this section about Fritz Haber. So remind me again who Fritz Haber was.
Was he the German scientist that won the Nobel Prize for nitrogen fixation?
He is the scientist, the German scientist who won the Nobel Prize in chemistry for developing nitrogen fixation. And so here in chapter 2, they begin to talk about the importance of nutrients in growing crops. And nitrogen is one of those nutrients that is usually limiting in soil. And when we talk about a nutrient being limiting in soil, it means that it's in low enough concentrations in soil that plants can't grow well without it, typically. And so one of the easiest ways to increase your crop productivity is by amending the soil, adding things to the soil that will increase the fertility of the soil, i.e. the nutrient content of the soil. More nutrients in the soil means more nutrients available to the plant, which means that your plants grow better, they grow bigger, they put on more seeds, more fruits, whatever it happens to be. If you go to the farm store or to the hardware store and you buy fertilizer, it will generally have 3 numbers, and the 3 numbers correspond to the amount of nitrogen, the amount of phosphorus, and the amount of potassium that are in the fertilizer. The NPK ratio, how much nitrogen, phosphorus, and potassium are in the fertilizer, because these are all nutrients that tend to be limiting in soil. And if you add them to your soil, you will get healthier plants. Some kinds of plants require more of one than the other, and so you can buy fertilizers that have different ratios of these things. And so corn, for example, is a high nitrogen demanding type of plant. And so amending your soil with additional nitrogen helps your corn along quite a bit. Some of you last time mentioned the fact that farmers do crop rotation, where they plant corn some years and soybeans other years. Soybeans are legumes, and legumes have associations with nitrogen fixing bacteria. And as a result of that, when they plow those plants under, they're actually enriching the nitrogen content of the soil because of those nitrogen fixing legumes. So nutrient cycling is important for agriculture. So it's important that you understand some things about nutrient cycles. So the first thing that I want you to be able to do is be able to recognize the major biological molecules. So if I show you a molecular model on a test or anywhere else, you would be able to look at that and say, oh, that's a carbohydrate, or that's a lipid, or that's a nucleic acid, or that's a protein. And I'll give you some key identifiers for how to do that. I want you to be able to know two of the nutrient cycles in detail, the carbon cycle and the nitrogen cycle. If you were in a biology class, we would also include phosphorus and sulfur in there. I'll talk a little bit about the phosphorus cycle, but not in a great deal of detail. Define and describe the inputs, outputs, and transformations or fluxes of carbon and nitrogen. And what we're going to do is we're going to start with carbon. We're going to build carbon just from our kind of general knowledge of how organisms work. and show you how you build a nitrogen, or how you build a biogeochemical cycle. And then we're going to take that method of building and apply it to the nitrogen cycle, which is a much more complicated, more difficult to figure out cycle. So when we get to nitrogen, we're going to talk about nitrogen fixation that is done by bacteria in association with plants, how that benefits the plants, and what limits there are to nitrogen fixation. Describe the difference between the types of fungi that occur in association with plants and how plants benefit from these associations with mycorrhizal fungi. That has more to do with phosphorus uptake than it does have to do with nitrogen uptake, but we'll talk about that. And then we'll ultimately want to draw a model of the nitrogen cycle at polyphase farms using the organisms at polyphase farms that they actually grow there. And so in the process of doing this, this will also serve as a bridge to food webs, which we'll cover right after we get back from taking the test. So food webs are on the schedule for the day after you give your genetically engineered organism presentations. All right. So why do plants and animals need nutrients? Plants and animals need nutrients because these nutrients are the physical building blocks of making you what you are. You are made up of molecules, and these molecules are made up of carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus for the most part. So we're going to start with the simplest of these, and we're going to build up in complexity. So the simplest of these molecules are sugars. And in this case, I've shown you 3 6-carbon sugars. And these are all different structures, even though they have 6 carbons, they have the same number of oxygens and the same number of hydrogens. The general formula for carbohydrates comes from the name. It's a carbon with a water molecule. So all carbohydrates have the form CH2O. And so it's carbon, 2 hydrogens, and an oxygen. So we have glucose, which is C6H12O6, because it's just 6 of these units that we have. But depending upon how the molecules are structured, they have different 3-dimensional shapes in some cases. And so, and they have differences in where bonds occur. And so here we have glucose, which has a hydroxyl group here and a hydroxyl group here. But in galactose, there is no hydroxyl group here. It's shifted its position. And because these bonds are fixed in space, the hydroxyl here versus the hydroxyl here gives it a different structure. So fructose is a 5-carbon sugar. So we have 1, 2, 3, 4. Oh, I'm sorry, it's 6-carbon sugar also. 1, 2, 3, 4, 5, 6. A 6-carbon sugar, but once again, it forms a 5-membered ring rather than a 6-membered ring because this extra carbon is hanging off the end here. So these are all C6H12O6, but they have different forms because of the specific differences in how those bonds are put together. Cellulose is down here. It's just a bunch of glucose molecules all bonded to one another. And in this case, you can kind of see how this structure is written differently from this structure. And you can see that the parts of the molecule, the hydroxyl groups and the hydrogen groups, either stick up above or down below this disk of the 6-membered carbon ring here. And so that's where you get these different types of 6-carbon sugars is in the details of where these hydroxyl groups are coming off and what direction they're coming off. Normally, we think of bonds as being freely rotatable around the bond axis, but in the case of carbon, which has 4 bonds, if carbon is bonded to 2 bonds that are part of a ring, you can no longer freely rotate that thing because it's part of a ring structure. So these are all carbohydrates, and so you should recognize carbohydrates as counting up the carbons: 1, 2, 3, 4, 5, 6. Hydrogens: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Oh, I missed one up here because that's H2. So there's 12 hydrogens, and there's 1, 2, 3, 4, 5, 6 oxygens. That's a carbohydrate. Cellulose is a long chain of glucose molecules that are bonded together, and those then are held together with hydrogen bonds that connect these two groups of hydroxyl groups here. And so you have these long chains that line up with one another, and then the long chains are bonded to one another. Have I talked to you guys about hydrogen bonds yet? Anybody remember that?
I don't remember.
Okay, well, let me just go through it really quickly. I did talk about it back when we talked about DNA, because hydrogen bonds hold the nucleotides of DNA together in the middle to create the double strand structure. In something like water, which is a hydrogen attached to an oxygen, which is then attached to another hydrogen, these are paired electron orbitals. When you look at the periodic table of the elements, these molecules, or these atoms up here, which are things like oxygen and sulfur, these things are what we call highly electronegative. What that means is that they tend to attract electrons to themselves. So if we were to draw the structure of oxygen, oxygen has 2 electrons in its first orbital, and it has 1, 2, 3, 4, 5, 6 in its other orbital. So oxygen has 8 electrons. And when we draw a Bohr model of the atom that looks like this, where we have the nucleus of the atom, which has 8 protons and 8 neutrons, that then implies that there are 2 electrons in the first shell and 1, 2, 3, 4, 5, 6 electrons in the next shell. And these are just one way that Niels Bohr came up with of envisioning what an atom is like. And so normally electrons like to exist in pairs, and the unpaired electrons are available for bonding. So hydrogen has 1 electron, and so it shares that electron with oxygen. So oxygen can fill up this orbital, and hydrogen can fill up its first orbital. Same thing with this hydrogen over here. They will share that electron with oxygen. Now, all of oxygen's outer shell is filled, and all of these 2 hydrogens' outer shells are filled. And so these bonds that are from electron sharing are called covalent bonds, because the electrons aren't lost by the atom that possesses them, they're just shared. But in the case of oxygen, oxygen is up here and is one of these highly electronegative atoms. And what it means is that it tends to pull electrons towards itself. And so when hydrogen shares its electron with oxygen, it tends to get pulled further away from hydrogen than it would normally be if hydrogen weren't interacting with oxygen. So a different way of thinking about oxygen, hydrogen in this situation is oxygen, the nucleus of oxygen is here, the nucleus of hydrogen is here, and the nucleus of hydrogen is here. And rather than thinking of these electrons as being stationary and trapped in these little orbitals, as chemists refer to them as, electrons are actually spinning around in space. And you never know where an electron is at any given moment. It's just spinning randomly around in space. Same thing is happening around hydrogen. Same thing is happening around this hydrogen. Their single electrons are spinning around in space. So sometimes you'll see in chemistry books, or actually pretty often you'll see in chemistry books, people talking about electron clouds. The cloud of electrons is just the physical space in which these electrons are spinning. It's a 3-dimensional space, and they're spinning around the nucleus. Now, when hydrogen comes into contact with oxygen, what oxygen tends to do is it tends to pull that electron closer to oxygen and pulls it away from the nucleus of hydrogen. Tends to pull this electron away from the nucleus of hydrogen and towards the oxygen. When it does that, the hydrogen is no longer fully balanced. It doesn't have a proton and an electron. It has a proton and a portion of an electron. So it becomes more partially positive. And so, and because this oxygen is pulling these electrons away from hydrogen, it takes on a more negative character. It's not a full charge like when you have sodium and chloride. So chloride is way over here on oxygen, fluorine, and chlorine. Chlorine is even further right in this diagram. And so when sodium interacts with chlorine, and sodium is way over here, the electron on sodium actually gets transferred onto chlorine, and this becomes a chloride ion. It takes on a full negative charge, and because sodium has lost its electron, it's missing one electron, so it takes a full positive charge because it now has one more proton than it has electrons, and this one has one more electron than it has protons. At that point, these two ions, a sodium ion and a chloride ion, will bond together because they actually have negative charges. They have opposite charges. In this case, what you don't get is you don't get the transfer of a full charge because oxygen isn't strong enough to totally strip hydrogen of its electron, but it does pull hydrogen's electron away. So what we get is we get a partially positive hydrogens with a partially positive or a partially negative oxygen. So when we write water in this way, we have to recognize that this hydrogen is partially positive, this hydrogen is partially positive, and this oxygen is partially negative. But what that does in water is it allows water molecules to interact with one another such that this partial positive hydrogen will form a hydrogen bond between that hydrogen and that oxygen, which then links this water molecule to the next water molecule. And so hydrogen bonds are intermolecular forces. They go between molecules. Because they're relatively weak bonds, they're oftentimes relatively easy to break. All you have to do to DNA to get it to separate into its two strands is to add a little bit of heat, and it will break those bonds, which then will split the molecule apart. But they're intermolecular forces, and the intermolecular forces are brought about by what we refer to as the polarity of these molecules. So these molecules, like water, are polar molecules, and it's because they have a positive pole and a negative pole. And so polar molecules will tend to make hydrogen bonds. And I did all of that to explain to you that these chains of glucose that go into making cellulose are held together from one chain to another chain by hydrogen bonds. And usually hydrogen bonds in diagrams are shown with dotted lines. Questions about that so far? When I ask you if you have questions, you should tell me because I'll go back and clarify anything. All right. So that was carbohydrates. So we have four classes of biological macromolecules, large molecules. The first of these was carbohydrates. The second of these are lipids. Now, another way of thinking about these different molecules is you can think about what are the atoms that go into them. So carbohydrates just have carbon, hydrogen, and oxygen for the most part, in the simplest of all worlds. Lipids also have carbons, hydrogens, and oxygens, but I'm going to add a P in parentheses there. But these carbons hydrogens and oxygens are found in different ratios. In lipids, what you find is you find long chains of carbon with a lot of hydrogens around them. Then these are bonded to what's called a carboxyl group. And then these are bonded to a glycerol, this carbon, carbon, carbon with the hydrogens around it. This carboxyl group is a carbon single bonded to an oxygen and then double bonded to another oxygen. So there are always 4 bonds around carbon. One of these goes to the long carbon hydrogen chain. One of these goes to a double bond to oxygen, or 2 of these go to a double bond of oxygen, and 1 of these goes to a single bond of oxygen. And so this glycerol molecule holds together 3 of these fatty acid molecules bridged between them by this carboxyl group. And so you have these long chains of carbons with hydrogens around them. You have a carboxyl group and you have the glycerol molecule. Now, sometimes you see double bonds between the carbons. When there's a double bond between the carbon, it means that each of those carbons has 1 less hydrogen associated with it. And you can sprinkle double bonds throughout these chains. And what it does is it changes the 3-dimensional structure of the chain. When you go to the store and you buy saturated fats versus unsaturated fats, or you see those things in nutritional labels on foods, that refers to how saturated this carbon chain is with hydrogens. If a carbon chain has all of the hydrogens that it can possibly hold on it, that's a saturated fatty acid. When there are double bonds such that you don't have the maximum number of hydrogens that you could have, that's an unsaturated fatty acid. And so these things form fats and oils, but then also the other thing that these things form is they form the membranes that go around all of your cells. But the lipid that is the one that is found in the membranes of your cells is a phospholipid. It's got a lipid where the glycerol molecule is attached to 2 fatty acid molecules, and then the third portion of the glycerol molecule is attached to a phosphate. This phosphate molecule has a full negative charge, and so as a result, this then becomes kind of a charged molecule itself. It has a neutral fatty acid end of the molecule and a negatively charged phosphate end of the molecule. So when I have a P in this list of atoms that are found in lipids, normally lipids are just carbon, hydrogen, and oxygen, but phospholipids, which is a very important molecule because it surrounds all cells of all organisms, that phosphate gets added in there. And it's an important phosphate because it imparts particular characteristics to the cell membrane. If we think of fatty acids, kind of phospholipids as they're illustrated in this figure, they have a ball on one side and two fatty acids coming down, a ball with 2 fatty acids, 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 and they're lined up in what we call a phospholipid bilayer. And this bilayer makes up what is the cell membrane that goes around all of our cells. Now, our cells are semi-permeable, which means that some substances can go through and other substances cannot. And because there's a negative charge on the outside of this cell membrane, anything that has a full positive charge is going to get stuck, either on the inside or the outside, and not be able to pass through. But neutral substances that don't have a charge will enter here. They'll come in contact with the neutral fatty acids in the middle, and they'll just slide right through because there's no charge that sticks them up in going through this. Things with a negative charge will be repelled away from the membrane, and things with a positive charge will get attracted to the membrane and kind of get stuck on the membrane. And so the function of membranes in cells is very much tied to the structure of this phospholipid. So this molecule is fairly important for the function of the cell membrane. So we've talked about proteins already, but we'll revisit this. We talked about proteins when we talked about proteins being the outcome of the central dogma, where we take DNA, we transcribe that into RNA, and then that messenger RNA is used to be translated into an amino acid sequence. And so, but we haven't talked in a huge amount of detail about amino acids themselves. So an amino acid is made up of an amino group, which has nitrogen in it, a carboxyl group, which we've already talked about, which is this carbon double bonded to an oxygen. And then there's a central carbon here that has a side chain on it. And this side chain is variable. And so if we go 2 slides later and look at these things, what we see is that here's the carboxyl group, here's the amino group, and here is the side chain. Carboxyl, amino, side chain. Carboxyl, amino, side chain. Carboxyl, amino, side chain. So what makes the different amino acids isn't the carboxyl and the amino group, it's actually the nature of the side chain. And in some cases, the side chain is just a simple hydrogen. Where is that simple hydrogen one? Oh, it's right here. So this alanine is just a carboxyl group, an amino group, and a single hydrogen. So when we look at these things, the bond that strings a bunch of amino acids together to make a protein is formed between the hydroxyl group of one molecule and the amino group of another molecule. So here's the central carbon, here's the amino group, here's the carboxyl group, and then there's a bond that goes from this carbon of this carboxyl group to this nitrogen of the amino group. And so in this way, the amino acids string together into long chains to make up a polypeptide. And when the polypeptide gets big enough, we start calling it a protein. These things all get strung together in long chains because sometimes these amino acids have charges on them. You can get parts of the chain bonding through hydrogen bonding to other parts of the chain. So amino acids, or so proteins begin as this linear arrangement of amino acids, but then that linear arrangement of amino acids turns back on itself because different parts of the amino acid sequence bond to other parts of that same amino acid sequence. And then we get these complex globs of proteins where this amino acid sequence is all tangled up on itself, and they create these 3-dimensional structures. And it's the 3-dimensional structures of proteins that have a lot to do with what function that they have. So when we look at the amino acids, we see that some amino acids are positively charged, some of them are negatively charged, some of them are polar, such that they have partial charges and not full charges within the side chains. Some of them are not charged at all, which means that they're neutral in terms of their charge. And so all of these amino acids have different characteristics based once again on kind of what atoms are in them and how those atoms are arranged and whether or not those atoms carry a charge. But you'll see that we have a lot of carbons, we have a lot of hydrogens, we have some oxygen, we have some nitrogen, but occasionally we also have amino acids that have sulfur in them. Remember that in the periodic table, oxygen and sulfur are up in that electronegative group of atoms, and as a result, you see that sulfur is one of these polar molecules. And so when we look at proteins as a major biological molecule, they have carbon, hydrogen, oxygen, nitrogen, which is different from lipids, and they have sulfur in some cases, although sulfur is relatively rare. Questions about amino acids or lipids for that matter? I forgot to ask you about lipids. All right, so the last one is the one that we've spent the most time on in class before, which is the nucleic acids. Nucleic acids, remember, have a backbone of phosphorus and sugar, phosphate, sugar, phosphate, sugar, phosphate. And then they have these nitrogenous bases This nitrogenous base is hydrogen bonded to another nitrogenous base that makes the double-stranded form of DNA, connecting this sugar to that sugar in a phosphate sugar, phosphate sugar, phosphate sugar backbone. So we have phosphate sugar chains connected by nitrogenous bases. So one of the things that you should get out of this is that nucleic acids have carbon, hydrogen, oxygen, nitrogen, and phosphorus. The phosphorus mainly coming from the phosphate and the nitrogen coming from the nitrogenous bases. And none of the nitrogenous bases possess sulfur, and so there is no sulfur in DNA or RNA. So this is the double-stranded structure of DNA. This is the single-stranded structure of RNA. Once again, the difference is that there's an oxygen on this 5-membered ring here, which makes this ribose, as opposed to there is no oxygen attached to this 5-membered ring here, which means it's deoxyribose. It's missing an oxygen. And so ribose is the sugar, deoxyribose, which makes it the difference between D for deoxyribose and R for ribose in DNA and RNA. This is also a single-chained molecule, but it has the same relative structure: sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, and attached to each of these sugars is a nitrogenous base. So once again, similar structure, difference in the sugar, and a difference in that one of these amino acids, one of these nitrogenous bases is traded out. Thymine isn't in RNA, uracil is there instead. So why do plants and animals need nutrients? Well, they need carbon, they need hydrogen and oxygen, but hydrogen and oxygen are super abundant. You find hydrogen and oxygen everywhere. They need phosphorus, they need sulfur, they need nitrogen. And so when you think about the atoms that organisms need, they need carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Sorry about that. I normally turn my ringer off before I go to class. So these are the big things. We're going to talk mainly about carbon and nitrogen. We'll talk to a lesser degree about phosphorus, and we won't talk at all about sulfur. If you're interested in those things, go take an ecology class. So we're going to concentrate mainly on carbon today. We'll talk about nitrogen later, and we'll mention phosphorus towards the end. All right. Questions about any of these molecules? On a test, I will give you a molecule, an image of a molecule, and I will say, which of the 4 biological macromolecules is this? And you'll need to write down either carbohydrate, lipid, protein, or nucleic acid. And I just got sour face from Mary about that assignment. This is why people don't turn on their videos, because I can then see their faces. So they're there if you have trouble remembering the key features. Once again, nothing but carbon, hydrogen, oxygen. But there's a set ratio, C, H, 2, and O. Here, carbon, hydrogen, oxygen, but a lot of hydrogens and carbons relative to oxygens, and there are these special features, the glycerol, the carboxyl, and these long carbon chains of fatty acids. Proteins have amino groups and carboxyl groups and a central carbon atom with a side chain on it. And these side chains vary a lot from one amino acid to another, and they get strung together in these long chains that then turn back on themselves. So fundamentally different structure than either of these two. And then the only long chain-like molecules that you have are nucleic acids that are similar to proteins, but they're very different in that you have the phosphate sugar, and the nitrogenous bases added to them. So learn the key features, and then you should be able to recognize the molecules. So the last chapter of your textbook, which I asked you to read for the day, today talks about ecosystems. And an ecosystem is just all the organisms in an area and all the biotic and abiotic factors that might influence those organisms there. So we have squirrels and trees of various species, we have insects, et cetera, et cetera. But the ecosystem is all that biotic stuff, all that living stuff, but it's also all the non-living stuff. It's the soil, it's the water, it's the sunlight coming, it's the wind, you know, everything that is in there. Today, we're going to be concentrating on the material aspect of the ecosystem. And the material aspect are the atoms that are flowing through this ecosystem. And so there are things like nitrogen, phosphorus, and sulfur, and carbon. And these substances, these material substances, exist in pools. You, yourself, are a pool of carbon, hydrogen, oxygen, nitrogen, sulfur, iron, some other trace elements, sodium, calcium, chloride. You possess all of those things in your body in different ratios. You are a pool. It's residing in there. It's not going anywhere. Now, when you go to the bathroom and you go pee or poop, some of the nitrogen and the phosphorus that is in you gets released to the sewage system. And in this case, that's a flux. It's the movement of the substance from one pool to another pool. And in this case, the pool that it's ultimately going to is water, actually, in the ecosystem. And so pools are where substances are stored. Fluxes are the flows of materials from one pool to another. And the cycle is the collection of all the major pools and all the major fluxes. So if we think about a generic nutrient cycle, we have pool 1, we have pool 2, and nutrients can move from pool 1 to pool 2 via flux 1, or they can move from pool 2 back to pool 1 through flux 2. Now, not all fluxes are dual direction like this. In some cases, fluxes go, well, in many cases, fluxes only go one direction. And so you might only have flux 1 going from pool 1 to pool 2, and flux 2 doesn't exist. We can add a lot of pools and a lot of fluxes. Here we have a pool of a nutrient. Here we have another pool, a third pool, a fourth pool, and a fifth pool. There's a flux, a unidirectional flux, a movement of substances from pool 1 to pool 2, a unidirectional movement of fluxes from pool 2 to pool 3. Pool 3 nutrients flow into pool 4, but then pool 4 fluxes back into pool 3, so that we have this, what we refer to as intra-system cycling, movement of pools back and forth, movement of materials back and forth between two pools. And then there is a flux 5 going from pool 4 to pool 5. We can actually physically say where these things are happening. Let's say that within this box, this is all stuff that happens in soil. Pool 1 is an atmospheric pool, and pool 5 is a water pool, so a stream or a lake or the ocean or something like that. Then we see a nutrient that begins in the atmosphere, gets transferred from one organism to another organism in these pools, can be recycled within soil, moving from pool 3 to pool 4 and back and forth, but then ultimately gets lost to soil in something like stream flow. And so this generic nutrient cycle, we can actually contextualize within a particular ecosystem. Atmosphere moving into soil or organisms, and then leaving the cycle through an output from the cycle, from the system, in the form of, for example, stream flow. So this is just generally how we think about nutrient cycling. Simple, more complex, contextualized within like a real ecosystem. Does that make sense to you? Okay. Now we're going to apply this to the carbon cycle. So when we think about the carbon cycle, you're already familiar with the equation for photosynthesis. 6 carbon dioxide molecules, 12 water molecules, add chlorophyll, rubisco, and energy from the sun, and you get glucose, you get 6 molecules of oxygen, and you get 6 molecules of water as byproducts of this process. You need energy to go into this to build up this complex molecule from these simple molecules. Once that's formed, though, we use this molecule to harvest energy. So that's a stored form of energy, and we're going to want to release that energy. So we have a glucose molecule. When we combine that with 6 molecules of oxygen, we'll get carbon dioxide and water as the byproducts, but we'll also get the generation of heat, and heat is just the energy that was pent up in this glucose molecule being released. This top equation is the equation for photosynthesis. The bottom equation is the equation for cellular respiration. So photosynthesis is how solar energy gets captured. Cellular respiration is how that captured energy is released and used. Okay. And notice that I put cellular respiration out here in front because we also use respiration to refer to breathing. And I'm never going to use respiration to refer to breathing. When I refer to breathing, I say breathing. So cellular respiration is this process of breaking down glucose to release the energy that is contained in the bonds of glucose so that the organism that is using that glucose can get energy. And so these two equations are basically backwards of one another. Start with carbon dioxide and water to get glucose. We need energy input to form those bonds in glucose. And we take glucose, break it apart to release the energy in those bonds, and we get carbon dioxide and water as a byproduct. So what we'll do at the beginning of class on Friday is we'll talk about the carbon cycle. So we'll talk about where carbon dioxide and glucose are formed, where they are in the ecosystem, and how carbon dioxide moves around in these large systems, because we are now out of time. Any questions? All right, then I'll see you on Friday. If you have any questions between now and then, let me know.